Okay, there we go. So like I was saying before, we were discussing a couple things in regards to some of the components of what a neuron is. We talked about the pumps, we talked about the different types of transport proteins, the channels. So we kind of understand some of the components now that are going to be found in these neurons. I need to talk about a couple of concepts because then we'll start to get in how we generate a nerve signal here. So this kind of brings in a little bit of physics into the discussion. We're going to discuss what Ohm's law is. So we have these transport proteins that will allow these different ions to diffuse across in and out of the cell, across the plasma membrane there. And so on a physics standpoint, okay, we're going to bring in a little bit of a physical science here. Right? When you get charged particles in motion, you will generate an electrical current, right? Electrical energy. And so we're going to see how our neuron is able to do that. Okay, so we're going to use a concept or a scientific law called Ohm's law. The lab test, it depends. It should be pretty much all labeling. And so I would say definitely yes. Make sure that I would use the Quizlets and the mock exams, the practice exams to kind of test your knowledge. I always just use my atlas. That worked for me. I would just keep going over the images that were labeled. That's usually what worked best for me. But then you can test to see if you're studying right, take the practice exams and see if that helps you. So Ohm's law. Okay, we're going to discuss it first here in a moment. All right, but when we're talking about Ohm's law, we're going to discuss some of the concepts, all right, that you need to understand. First, what is voltage? What is current? What is resistance? All right. Voltage is going to be when we see the difference, and we were discussing this before when we were talking about the electrical gradient there, right? When we were talking about membrane potentials, right? But when we talk about voltage, that's going to be a difference in the electrical charge, all right, between two places. And we're going to use, since we're talking about the neuron, we're going to discuss that and measure that. You're absolutely welcome, Rachel. We're going to measure that using millivolts. Okay, so when we're discussing what a current is, that's going to be the actual movement of those charged particles. Like in the wires in your walls, when the electrons are moving through, it's generating a current. So these particles, when they're moving across the plasma membrane through those channels and those pumps there, we are going to see, all right, the current being utilized. And so we're going to use that current, all right, to perform certain physiological functions here. And then finally, resistance. Right, that's going to be the opposition to the movement of those charged particles. Okay, in our case, it's going to be the actual physical barrier of the plasma membrane. And so when we have an increase in resistance, right, it's going to be more difficult for charged particles to move. So we'll also see a decrease in the current here. So on our next slide here, all right, you can see what Ohm's law is. It's current equals voltage over resistance. Right, so we have our little formula here for it. Okay, so you'll see, and again, I'm not going to try to make you a math genius here when we're dealing with fractions and this type of formula here, but we'll see that there's going to be an inverse relationship between some of the components of Ohm's law. So one of the things that we see, okay, if we increase our current, which is the I value here, if we increase current, all right, we will see one of two things or both things. We'll see larger voltage and we'll see smaller resistance. So how does this all apply to the neurons? Okay, well, all right, when those charged particles are moving across the plasma membrane, we are going to generate some sort of current there. And so the voltage that we're going to see, how the outside of the cell is positive, the inside of the cell is negative, right? We're going to see, all right, that voltage there, that's going to be due to the unequal distribution of ions. Well, what is that, Dr. Kaz? I've got one phrase for you, all right? Banana floating in the ocean, okay? That's what is going to tell us what our unequal distribution of ions. We have more potassium ions inside the cell, all right? And we have more sodium and chloride ions outside the cell. And so they're always trying to flow down their concentration gradient. That's going to be our voltage. All right, and then finally, our resistance, all right, what's going to resist the movement or prevent the movement from the ions is the plasma membrane. Okay, so how do we change that resistance? Okay, because we know that the ions can't directly diffuse right through the plasma membrane. They're charged. They're not small and nonpolar. Okay, so that's a problem for us. So we have to put these transport proteins in the plasma membrane, okay, these channels. And so what we'll see is, okay, when those channels are open, that will decrease the resistance. Well, if you also want to decrease the resistance, you can increase the number of channels. So when they open, it'll flow through. All right, so here's a great slide here discussing what Ohm's law is and kind of puts things into perspective a little bit. Okay, so now you can see our neuron here. And so when we're measuring the voltage, we're totaling up, all right, the difference in the electrical charge between what's outside the neuron and what's inside. So the sodium chloride and some of the other ions that are outside, and then the potassium on the inside, but also, all right, we notice that's a positive ion, but we have our negatively charged phosphate ions and our negatively charged proteins. All right, they're going to help to really contribute to that negative charge on the inside. All right, so our plasma membrane offers the resistance, all right, prevents or tries to slow down, all right, opposes the movement of the charged particles across the plasma membrane. And then you can see our current, what's generating our current, the movement of these charged particles, these ions in and out of the cell. And that's going to create our current. So what is the electrical gradient? An electrical gradient is the difference in the electrical charge between two areas. That's going to be our voltage. Then our chemical gradient is the unequal distribution of the substance between the two areas. That's our banana floating in the ocean, okay? So, perfect place to kind of take a breath in and blow it all the way out. Now we're going to take all this information that I've kind of piecemealed together and presented to you, and now we're going to fit it into our story. And so we're going to start off with our neuron at rest, when it's not doing a darn thing. Okay, this is what happens, all right, in a decent number of nerves in your body, okay, and the neurons that make up that nerve when you're, let's say, sleeping at night, okay, or if you're in a coma or something. So what we're going to see, right, with these resting neurons, okay, we're going to see a difference in the distribution of certain substances across the plasma membrane. So what creates that difference? Well, that is going to be due to pumps, our transport proteins. Remember, the sodium potassium ion pump, right, is going to pump sodium out of the cell. It's pushing it out of the cell against its concentration gradient, and it pushes potassium into the cell against its concentration gradient. So we'll see much more potassium in the cytosol, all right, compared to the interstitial fluid, and we'll see more sodium chloride and calcium in the interstitial fluid than we will see in the cytosol, because the other type of pump that we also see in the plasma membrane, but it's only found down in the transmissive segment, which is the synaptic knobs, is the calcium pump, and that pumps calcium out of the cell and into the interstitial fluid. Okay, so we have this uneven distribution of these substances due to the actions of the pumps. Now, that's happening. Then we have our gated channels. And what did we say about gated channels? They're normally closed. And so when we have a neuron at rest, these channels are closed, which means that whatever they're supposed to allow passage of is not moving through there. So because of this situation, the outside of the neuron is going to be more positive, because we have all these positive cation ions outside of the cell, okay? And so on the inside of the cell, we do have a cation ion or potassium, but we have phosphate ions that are negatively charged and our proteins that are negatively charged. And so they're going to contribute much more negative charges than the potassium ions that are inside the cell. So they'll one for one, they cancel each other out. One positive charge cancels out a negative charge. Okay, so there's more negative charges that'll cancel out all the positive charges of the phosphate. So the inside of the cell is going to have a negative charge to it. And so when we measure that charge across the plasma membrane, we get this value here, in which you need to burn this in to your memory. Okay, since the neuron is at rest, okay, we refer to the membrane potential as the resting membrane potential. So when I say resting membrane potential, you know that I'm referring to a neuron that is at rest, where all this is happening. And so when we take that charge, it's a negative 70 millivolts. So finally, we have a quantitative value here. All right, cool. I got my negative 70 millivolts. I know that that's the charge at the plasma membrane, okay, when the neuron is at rest. So here you can see what that looks like, okay, with a neuron at rest. All this stuff is happening, okay? You have these these channels here are located all throughout. And the pumps, you can see if we zoom in on these channels here, okay, we'll start with the receptive segment. Okay, in our receptive segment, we're going to have the leak channels, but we're not talking about them right now. We'll have the sodium, potassium ion pump, okay, but we'll have chemically gated channels all throughout the receptive segment. They're all closed, okay? They're all closed. Then we get down into the initial segment and the conductive segment. We have voltage gated channels. They're all closed. And then we get down to the transmissive segment. We have our calcium channels, and they are all closed. Okay, so gated channels are going to be closed. All right, so now let's talk about, right, we're still talking about a neuron at rest, but now let's talk about some of the substances, the ions, sodium, potassium, okay? We're going to talk about them and their role in how we get that negative 70 value for our resting membrane potential. So now I'm going to show you how we figure out this resting membrane potential, like what contributes, what makes it negative 70? How come it's not negative 90? How come it's not negative 60? Okay. So we're going to start off with the most important ion, right? And that is going to be our potassium. So the potassium ion, right, is going to influence the resting membrane potential the most. So we all know from our banana floating in the ocean that potassium wants to leave the banana. It wants to get out of the cell, okay? So it has the leak channels to do that. So it's just going to leave, okay? Meanwhile, the sodium potassium ion pump is going to pump sodium in, potassium back into the cell, but it'll just leak out again, okay? But we want it to leak out because that's what's going to help to generate this resting membrane potential. So what causes potassium to leak out? It goes from an area of high concentration to low concentration, so it moves out of the cell. So as long as that concentration gradient is present, it'll move. If it's equal, if it reaches equilibrium, which there's just as much potassium outside the cell as inside the cell, it's not going to move. But now let's not forget, potassium is a positively charged ion. And so as it's diffusing out, what did we say the inside of the cell was, charge-wise? Look at our picture. Check it out. Positive outside, negative inside. Okay, so that positive ion, okay, is diffusing down its concentration gradient, but that's not the only thing that influences how it diffuses. Remember, opposites attract? Okay, that electrical pull that's generated from those negative phosphate ions and the negative proteins inside is going to kind of tug on that positively charged potassium ion. It's going to say, hey, wait, come back here, come back to us. You're attracted to us. Okay, so this makes it a little bit more difficult for potassium to diffuse out. And now in the beginning, the concentration gradient is so large, it just starts to diffuse out. But as the concentration gradient becomes less and less and less, and as we get closer to equilibrium, well, then the electrical pull, the electrical gradient is going to play a larger role on that potassium. And so it's going to slow down its ability to diffuse out. And so at a certain point, we're going to reach equilibrium. And that's when we have that chemical concentration gradient, okay, is going to be equal to the pull that those negative items inside the cell have on that positively charged potassium. Okay, so because of all this, right, and we're talking about with the leak channels and whatnot, okay, if potassium were the only molecule, say there was only potassium leak channels, no other type of leak channels. If that were the case, our new resting membrane potential would be at -90. So if we just had potassium leak channels, okay, because of the concentration gradients, the chemical concentration gradient, and the electrical gradients, right, that equilibrium would be at -90. Okay, but that's not what we see as our resting membrane potential. So now, because there's some other things that are involved. Okay, let's talk about those other things. Sodium leak channel, or sodium, okay? So we did talk about how sodium leak channels are present, but there's not as many. Potassium leak channels far outnumber the sodium leak channels. So sodium will also play a role in determining what our resting membrane potential is going to be. But in this case, banana floating in the ocean, sodium's on the outside, and it's trying to get in. So it'll flow through its leak channel, okay, because again, it's going to flow down its concentration gradient, and there's far less of it inside the cell, so it just floods in, and it's getting pulled in by the electrical gradient generated from those negative proteins and those negatively charged phosphate ions. So it's not being hindered at all. In fact, it's begging to be invited. Come on in, flood into this cell. So that's what it does. But again, because there's so few sodium leak channels, okay, it doesn't influence the resting membrane potential all that much. But because it does influence it, now our resting membrane potential is around -70. If it wasn't for these sodium leak channels, then our new resting membrane potential would be -90, and that's because of potassium's movement. You need to know that. Okay, so also at our resting neuron, we got those sodium potassium ion pumps. Remember, they're always pumping, they're always working. Okay, and as long as there's sodium inside the cell that can get pushed out, they're going to be working. And as long as there's potassium outside the cell trying to get in, it's going to be working. So what this structure does, this transport protein, it pumps 3 potassium ions out of the cell. And did I say, 3 sodium ions out of the cell while pushing 2 potassium ions into the cell. And so that is going to contribute about 3 millivolts, very, very slight. Okay, so you can see it doesn't really play that much of a role in establishing the resting membrane potential. Okay? It does a little bit, okay, but not nearly as much as sodium and not even close to how much potassium does. So it does play a small role in establishing our resting membrane potential. But here's the thing that I want you folks to know. This is hugely important. Okay, it maintains the concentration gradient. It's making sure that there's more sodium outside the cell and more potassium inside the cell. So there is an existing concentration gradient so those ions can diffuse. And in fact, it's plays such a big role in that. Check it out, 66% of a neuron's energy expenditure comes from just the operation of these pumps. 66, or 2/3 of a neuron's energy expenditure comes from these pumps. So that's pretty important, okay? So they play a big role. So here you can see what's happening, all right, at a resting neuron. Okay, so here's our sodium potassium ion pump pushing out, all right, the sodium ions and pushing in the potassium ions. You got leaking channels of both sodium and potassium. Far more, look at all these potassium channels compared to the one sodium. And then down inside the cell, okay, we have our inorganic phosphate molecule and our negatively charged proteins, okay, which are going to generate that negative charge inside the cell. So all this is going on, and it's just happening. So we decided to take a voltmeter and stick the leads outside the cell and inside the cell, and that's the value we came up with. -70 millivolts is how, all right, this resting membrane potential reads on a voltmeter. Okay, So now, okay, here you can see our resting membrane potential on our gated channels. Again, -70. And what are all these gated channels doing? They're closed, 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 closed. Okay. Sodium channels, potassium channels, and our chloride channels, those are closed when the neuron is at rest. Okay, so as long as you have that picture in your head, because now we're going to talk about how we're going to disrupt that. Okay, so the resting membrane potential is primarily established by what two structures embedded in the plasma membrane? The resting membrane potential, okay, the -70 millivolt value. What are the two structures? Okay, the sodium leak channels and the potassium leak channels. Primarily, though, the potassium leak channels, right, will influence that value of the resting membrane potential way more than sodium will. Okay, so now you know what a resting membrane potential is. Now you know what's going on at a neuron when it's resting. So let's disrupt that. Let's change that membrane potential. So let's change that resting membrane potential. And to do that, we're going to introduce to you a couple terms here: depolarization, repolarization, and hyperpolarization. So in order to change that membrane potential, we need a stimulus. And when that stimulus occurs, then we're going to see the opening of some of our chemically gated channels and some of our voltage gated channels. And when those open up, okay, now we're going to change, okay, the ion flow across that plasma membrane. Okay, so what we were talking about before, okay, now we're going to influence that. And when that occurs, then we're going to change our resting membrane potential. All right, we can make it more negative, make it maybe -77, or we can make it less negative, make it like -66, -55. Okay, so we're going to talk about that. So let's define a few terms here for you. The first one is depolarization. This only occurs at the plasma membrane, but here's the definition, and you need to burn this one into your memory. Inside of the cell becomes more positive than the resting membrane potential. What's our resting membrane potential? -70 millivolts. You should know that. And so when we make it more positive, isn't that the same thing as making it less negative? So we go from -70 to -60, like the example here shows us. That's depolarization. We've made the inside of the cell more positive. We're going to talk about that. So what we did is we opened up some channels. And because of that, right, we would see, right, the inside of the cell becoming more positive. Well, how do we do that? Let's bring in some stuff that's positive that's outside the cell. Well, I like sodium. There's a lot of it outside. So let's open up a sodium channel. Sodium rushes in. It brings its positive charge with it, and the inside of the cell becomes more positive. So much so, right, we'll eventually reach this value here, +30 millivolts. And then those sodium channels will close. Boom. It seems almost as quickly as they open, they close. Okay, so let's talk about what we're seeing here. Here you can see our picture, right, of our gated channels. Now we've talked about depolarization. Let's make the inside of this cell more positive. Yes, less negative. Okay. So we'll open up a channel that's going to bring positive stuff from the outside into the cell. And these are our positive things. Sodium comes in, makes the inside of the cell less negative, -60 millivolts. That's how we do it. That's how we do this. Okay, so we depolarize the cell, we open up those gated sodium channels, and sodium comes into the cell. Okay, that's depolarization. So what's repolarization? You ever heard of the saying, what comes up must come down? Okay, so now we have to return our membrane potential that is now +30 millivolts. We got to get that back down to -70. Okay, I got this. we need to open up a gate. Okay, we've already opened up one. Let's see if we can open up another. How about if we open up the voltage-gated potassium channels? Okay, so that's what we do. We open them up, right? Once we hit this value of positive 30 millivolts, okay, these gates are closed. Once they get that value, boom, they open up. And guess what? Potassium is going to move across our plasma membrane from an area of high concentration to an area of low concentration. Banana floating in the ocean, that potassium inside of our cell will leave the inside of the cell. Guess what? Guess what? It takes its positive charge with it. So now the inside of that cell is going to become more negative. And we get back down or as close to our resting membrane potential of -70. All right, as this repolarization process occurs, remember these folks, the voltage-gated sodium channels? What did we say about them? They have three states. They have a resting state, which we just talked about. That's when they're closed. We have an activation state and an inactivation state. All right, so what happened during repolarization, all right, we will see the voltage-gated sodium channel go from the inactivation state to the resting state. The activation state occurs here. This is the activation state, depolarization. Both the gates are open. Sodium is moving through. Then once that sodium, once that sodium is moved through, okay, and we hit that positive 30 millivolt value, then the inactivation gate closes. And during this repolarization period, we go from the inactivation state back to the resting state. Okay, so here's our repolarization. And you can see here's a closed potassium channel. Here's an open potassium channel. Potassium then starts to leave the cell, taking its positive charge with it. And that's how we start to bring, all right, the cell that was once negative outside and positive inside, that's how we flip it back to being positive outside and negative inside. That's how we get our resting membrane potential back down to that -70 because potassium diffuses out. Simple, simple. All right, one more term I want to define for you is hyperpolarization. Okay, this is what happens when the inside of this cell becomes more negative than the resting membrane potential. More negative, you say, Dr. Kaz? Yep. Resting membrane potential is -70. What happens if it goes to -72, 75, 78, 80? Okay, that is more negative than the resting membrane potential. So this happens, okay, from the potassium channels, or we haven't talked about these guys, or our gated chloride channels. So what will happen is we're repolarizing the cell, potassium is leaving the cell. Sometimes those gates don't close right on track. They don't close at -70. They might close at -72, -74. Doesn't get it perfect every time. You also might see the opening of our chloride gated channels, and chloride is in higher amount outside the cell, and it's going to try to rush into the cell. Well, guess what? When it rushes into the cell, it brings its negative charge with it. It makes the inside of the cell more negative. So that's what we're seeing. So when we're talking about hyperpolarization, we can see it as a loss of the potassium ion, all right, leaving the inside of the cell, or we could see it as a gain of that negatively charged chloride ion. So if we get to this -70, all hope is not lost. We close these gates, no more ions flow across the plasma membrane. And so we're stuck at the -80 value, for example. Well, no problem. The sodium potassium ion pumps are going to take care of the issue by getting us back down to that -70. They're just going to pump sodium out of the cell and potassium into the cell. And that's how we're going to get back to our resting membrane potential. It's really a nice little kind of flow. So here you can see hyperpolarization. Our voltage gated sodium channel is closed, all right? But, all right, you can see our potassium ion channel is open and our chloride channel is open. So that negatively charged chloride enters in, making the inside of the cell more negative. And potassium leaves the cell and it takes its positive charge with it, makes the inside of the cell more negative. Okay, so it's either going to be hyperpolarization is caused by potassium flowing out or chloride coming in. How does depolarization and hyperpolarization occur in a neuron? Okay, so depolarization, we're going to make the inside of the cell less negative. Okay, so we're going to either bring in some sort of positively charged ion, a cation, or our voltage gated sodium channels open up and sodium pours in. And that positively charged sodium, right, will help to depolarize that cell. Hyperpolarization, now we're going to see when our gated potassium channels open and potassium moves out of the cell, taking its positive charge with it. Because hyperpolarization, we're making the inside of the cell more negative. Okay, so that could be one cause of hyperpolarization, or the gated chloride channels open up and chloride enters into the cell, bringing its negative charge with it, also making the inside of the cell more negative. Oh, all right, there's some big concepts here that we covered. And I gotta say, I am so proud of everybody for sticking with me this long, okay? This is some of the stuff that I love to tell, these storytelling issues or concepts. And we're gonna be moving into, I taught this material earlier today, and so I really enjoyed it. So I apologize if it seems I'm getting away from myself, okay? But I wanna kind of get back on track here and get into what now is going to happen at the different parts of the neuron to generate an action potential, a nerve signal, because that's what nerves do. They're excitable cells that have to conduct a nerve impulse. So now we're going to discuss how all the parts of that cell play a role in creating a nerve signal. So we're going to break it down by each segment of the neuron. There's 4 segments. So we're going to tell a story about what happens at each of these segments here. So we're going to start off with what happens at the receptive segment. That's the cell body and where the dendrites are. All right, and then we're going to work our way down all the way to the transmissive segment, which is where the neurotransmitters are going to be located that get released onto another neuron or an effector cell or effector organ. All right, so here you can see all the different segments. The receptive segment, cell body or soma, and the dendrites there, which are these short processes here that receive input. Then we have our initial segment. This is the ultimate goal here for what is going to happen here in the cell body that we're going to talk about here. But ideally, whatever happens in these dendrites or in the cell body, our main goal is to get, all right, this area here, right, to initiate an action potential. So I'm going to talk about what's going to happen here, right? But our ultimate goal, so we can generate an action potential, is to initiate that action potential in this area. We're going to talk about how that happens. Then once we get the action potential going, okay, it's going to go from point A to point B, all the way down what we call the conductive segment. That's the axon. And then as it travels down the axon terminals out to the synaptic knobs, then we finally wind up with that action potential, that electrical signal getting converted into a chemical signal, which is going to be the release of that neurotransmitter. So we're going to walk through this, and you're going to be amazed, and you're going to love it. I hope you do. All right, so let's start off with the receptive segment. And so the receptive segment is going to be the cell body and the dendrites, but there's not going to be an action potential that occurs in the receptive segment. The only place that an action potential can occur in is the initial segment, which is the axon hillock, and the conductive segment, which is the axon. Right? In the receptive segment, we have something different. We have what's called graded potentials. So they're like a smaller version, a smaller, weaker version of an action potential, somewhat like that. Okay, so we refer to them as small and short-lived changes in the resting membrane potential. So we're going to talk about how these graded potentials will hopefully get the generation and the initiation of an action potential to occur in the initial segment. All right, so how we start to generate these graded potentials is when we open these chemically gated ion channels that are found in the dendrites and in the cell body. So a presynaptic neuron is going to release its neurotransmitters onto our postsynaptic neuron. And the postsynaptic neuron has these chemically gated ion channels, which are normally closed. And so we're now going to talk about how that presynaptic neuron's neurotransmitters are going to influence, all right, certain chemically gated ion channels to open up. And if they do, they're going to hopefully generate some sort of change in that resting membrane potential. Okay, but here's the scenario though. In the receptive segment, these changes, these graded potentials do not last long and they're local. So I look at it like this. It's like throwing a rock into the water in a pond or a calm lake, and the ripples start to move outwards. But as they move out further and further away from the area that you threw the rock in, they become weaker and smaller. Okay, because they encounter, all right, those energy waves encounter resistance, which brings us back to Ohm's law. So when we open up these chemically gated ion channels, those ions will diffuse into the cell. And normally what they'll do is, all right, they'll travel parallel to the inside of the plasma membrane, like a ripple in a water, okay? But the further that those ions travel, all right, they become weaker and weaker because they encounter resistance. Where does that resistance come from? It comes from all the stuff in the cytosol. Okay, the cytosol is not like water, okay? So it's like swimming through, I don't want to say jello, but real thick oil, not honey. Okay, I'd say it's closer to oil. It's just harder. So it becomes weaker and weaker and weaker, all right, the further and further it travels from where it entered into the cell. Right, so what we need to do is, all right, we are going to be able to, with these graded potentials, all right, yes, they're small and short-lived. We need to make them big enough, okay, because we eventually need to trigger the initiation of an action potential in the axon hillock. All right, so these graded potentials will vary in degree, all right, how much they're able to change that membrane potential. And, all right, in what direction did they change that resting membrane potential? Because remember, they can either make the resting membrane potential, all right, more negative or more positive. So it can go from -70 to -75 or to -65. So it depends on what type of graded potential occurs here. And we'll discuss that. Right? So again, we'll see these graded potentials, large or small. And again, how does it determine if it's large? The more gates that you open up, the larger the potential, the graded potential. Okay? Fewer gates that open up, the smaller the graded potential. So there's two types of graded potential. One will cause depolarization, and the other one will cause hyperpolarization. And so we're going to discuss that. And again, before I forget, when we're talking about the opening of these chemically gated ion channels, remember, gated channels are specific to one type of ion. Okay? One type. Okay, so the receptive segment, our dendrites and our soma, cell body. This occurs, all right, here on the postsynaptic neuron. The presynaptic neuron is going to be the one that releases the neurotransmitter onto the postsynaptic neuron. So when we are talking about these graded potentials on the postsynaptic neuron, we refer to those as postsynaptic potentials. Remember, I told you there's two types. One causes depolarization, the other one causes hyperpolarization. So the postsynaptic potential that causes depolarization, we call that an excitatory postsynaptic potential, or an EPSP for short. So these type of potentials will depolarize the plasma membrane. Okay, it'll make it less negative. So the postsynaptic potential that causes hyperpolarization, okay, that's going to make the resting membrane potential or the membrane potential more negative. Those are called inhibitory postsynaptic potentials. So we're going to see, all right, how you can have, all right, when we're talking about these postsynaptic potentials, you can have hundreds of EPSPs being generated and hundreds of IPSPs being generated, right? And it depends on what neurotransmitter is being, again, released and what channels are being affected. So on our postsynaptic neuron, we can have hundreds of other neurons synapsing onto that one. And some of those will be generating EPSPs, and some of those presynaptic neurons will be generating IPSPs. So basically what we have to do is we have to add everything up. And what we'll do is it's like cashing out your chips at the end of a night of gambling, right? You're going to add up all the IPSPs and you'll add up all the EPSPs. Okay, and it's a one-to-one situation for canceling each other out. So one IPSP will cancel out an EPSP. And at the end of the night, we'll find out, do we have more EPSPs or IPSPs? And we'll talk about what happens there in a moment. So I want to talk to you about how we generate, how do we create, how do we make an excitatory postsynaptic potential, an EPSP. All right, so here's what we see. Our presynaptic neuron releases its excitatory neurotransmitter, and that neurotransmitter diffuses across the synaptic cleft. It binds on to the receptor on the postsynaptic neuron there, and it opens up a chemically gated cation channel. All right, now, we didn't say which channel, we said a cation channel. Well, we have two cations, right? We have sodium outside the cell, and we got potassium inside the cell. That's right. So it opens up those channels there. Sodium moves into the cell, potassium leaves the cell. But way more sodium moves into the cell than potassium leaving the cell. And remember, because sodium's getting begged to come into the cell. It's like, hey, we're negative in here, you're attracted to us, and you know, you're flowing down your concentration gradient. Whereas with potassium, yeah, potassium is like, all right, I'm leaving this cell, you know, because there's less potassium outside the cell. As it's trying to walk out and move out of the cell, the negative charge inside the cell from the proteins and the phosphates is kind of like, hey, it's kind of pulling on the potassium. Wait a minute, bud. All right, slow down, you know, don't leave. All right, you're attracted to me. Okay. So that makes it a little bit more difficult for potassium to leave the cell. But like I said, way more sodium moves in. So the inside of the cell becomes more positive. That's depolarization. We go from a -70 resting membrane potential to possibly -68, -67. Okay, that's good. Okay, we're depolarizing our cell. But problem is, this is a graded potential. It's small, and it's short-lived, it's localized. So this local current, that sodium that we have with the sodium, it becomes weaker and weaker and weaker as it moves, right, parallel to the plasma membrane towards the axon hillock. Okay, all of it is always trying to move towards the initial segment there. All right, so how do we determine the amount, all right, or the change in our resting membrane potential? You heard me say it went from -70 to to negative 68. Well, how come it didn't go from negative 70 to negative 60? Right? Well, the reason why is, okay, we didn't have as much neurotransmitter being released. So if you want to get a bigger change in that resting membrane potential, right, get more neurotransmitter out there and get it to stay on the receptor longer, because as long as it's on the receptor, the gate will stay open and sodium will move into the cell. Okay, so we've got to release more neurotransmitter, causes more cation channels to open up. Right, and then we can go from negative 70 to negative 65 to negative 60 instead of just a negative 68. So that's how we're going to do it. I love this picture. Real quick, just into words. I'm almost repeating myself, but yes, I am. All right, just to kind of go up, back up here to what an EPSP is. That's when we depolarize the cell. Okay. Keep that in mind. That's what we're going to do with EPSP. So here's our presynaptic neurons. They're releasing their excitatory neurotransmitter. That neurotransmitter binds on to all these cation channels, causes those channels to open up, causes sodium to move in, and causes potassium to leave. But more sodium enters, right? Then potassium exits. So we make the inside of the cell more positive. And so, of course, we start to generate this little current, and you can see it starts to move towards the initial segments. It wants to move towards the axon hillock. So when we're graphing this, here's our resting membrane potential of negative 70. Here's the stimulus, the opening, all right, of those chemically gated cation channels. And now our cations flowing across the plasma membrane, and we get a little blip. And that's about negative 68. So we made it, we depolarized it a little bit, but we didn't depolarize it enough yet. Because now we're going to soon talk about this right here. What's this? What's this threshold value? Well, for right now, I'll tell you that's the value that we need, all right, to get to, which is negative 55 millivolts. Okay, that's the value that we need to get to, all right, at the axon hillock, at the initial segment. We need to get the resting membrane potential, right, to change 15 degrees and convert that membrane potential to negative 55. And then this has to happen at the initial segment, and that will cause the propagation of an action potential. More on that, we'll get there. Okay, so that is a generation of an EPSP. So how do we generate an IPSP? Similar scenario, folks, similar scenario. So here's what happens, okay? Our presynaptic neuron releases its inhibitory neurotransmitter, okay, at the synaptic cleft. That neurotransmitter diffuses across the cleft, right, binds on to chemically gated potassium channels and chemically gated chloride channels, okay? Depends on what type of neurotransmitter is released and what channels are present there, right? But then remember I said certain neurotransmitters are going to stimulate certain chemically gated channels to open up. But regardless, right, if we open up the chemically gated potassium channels, then potassium moves out of the neuron. And it makes the inside of the cell more negative because potassium takes its positive charge with it. If that neurotransmitter opens up our chloride channels, then of course, chloride will flow down its concentration gradient into the neuron, and it will do the same thing. It'll make the inside of the cell more negative. And so we went from negative 70 to negative 72. We made it more negative. That's hyperpolarization. Same scenario. Okay, these ions, when they flow into the cell, they're going to move parallel to the plasma membrane, trying their hardest to get to the initial segments. All right, but they're going to encounter resistance. That's what happens. And so their signal becomes weaker and weaker. Right, and hopefully it can get there. But if not, all right, we'll try another IPSP. All right, so here you can see, when we're talking about IPSP, that has to do with hyperpolarization, and that has to do with potassium leaving the cell or chloride entering into the cell. So similar situation here. Okay, our presynaptic neurons are going to release their inhibitory neurotransmitter, right? If it is specific to the potassium channels, okay, it will cause the opening of the chemically gated potassium channel. Potassium will leave the cell, and the inside of the cell will become more negative, and those IPSPs will move towards the initial segment, encountering resistance along the way. Right? Or if that neurotransmitter is specific for the chloride channels, it stimulates the chloride channels to open up, and then chloride can enter into the cell, bringing its negative charge with it and making the inside of the cell more negative, and also hyperpolarizing the cell. And so that's what we're seeing here. Here is our -70 resting membrane potential. The stimulus occurs, the gates open up, the ions flow across the plasma membrane, and we've hyperpolarized our cell and made it more negative, past the resting membrane potential, moving us away from this threshold value that we hope to find out what it is one day. We will, I tell you, we will find out what this threshold value is in a moment. Okay, so when this is occurring, all right, we now know what an IPSP and what an EPSP, okay? In case you may have forgotten, what generates those, all right? What's responsible for those? The presynaptic neurons, okay? Those presynaptic neurons and whatever neurotransmitter they are emitting. So what we're going to see is, right, like I said, you can have the postsynaptic neuron have hundreds of other neurons, right, synapsing with it. And some of those will release excitatory neurotransmitters, and some of those will release inhibitory neurotransmitters. And this can be simultaneously occurring from all these different types of neurons here. All right, but what we'll see is, it could be all EPSPs, it could be all IPSPs, it could be a mixture of both. If it is a mixture of both, then that's when we have to start to do a little bit of accounting, and we have to start to cancel IPSPs out and EPSPs out with one another. Okay? So again, we're going to see, all right, this simultaneous release occur. And so this picture shows a great example. I love this electron micrograph slide here. And you can see, it's pretty cool. All right, over here on the right, you can see here's our postsynaptic neuron cell body here. And you have all these, look at all these telodendra, synaptic knobs, okay? And so some of these could be releasing excitatory neurotransmitters, and some of these could be releasing inhibitory neurotransmitters. But you can see all these different types of synaptic knobs, right, will vary. And so, but again, all the graded potentials, whether it's an EPSP or an IPSP, is going to move towards the initial segment. Our goal is to get, all right, these EPSPs and IPSPs here. This is where we're going to add everything up. So all these people are gambling at the casino, right? But when it's time to cash out, you got to come here. Okay, you got to come here, and we're going to see, right, if you have enough to generate an action potential. Enough what? Well, we're going to find out here in a moment. So how are EPSP graded potentials established in the receptive segment of the neuron? Okay, we are going to depolarize the postsynaptic cell. To do that, we're going to use excitatory neurotransmitters. They're going to open up those chemically gated cation channels. Sodium flows in, potassium flows out, all right? But as a result of that, we make the cell slightly more positive inside. We depolarize it slightly. Okay, so now the initial segment. So we know what's going on in the receptive segment. Okay, graded potentials are happening. Okay, and we're trying to change that resting membrane potential. We want to get to that threshold value that I was telling you about. Now I get to talk to you about it. Lots of stuff happening, folks. It's a beautiful thing. Lots of fun stuff. Okay, so I think in first grade nowadays, it might even be sooner, I don't remember, you learn about addition and subtraction. When they talk about getting the sum of something, that's adding everything together, and you get the sum of whatever it is. Well, at the initial segment, this is where we get the summation, the sum of all the EPSPs and all the IPSPs. This is where you go to cash out your chips. So what we're going to do is we're going to add up all the voltage changes that occurred in all the dendrites and the cell body, all the voltage changes, right? And we're going to add them all up. And we are going to see if the sum value of that reaches what we call the threshold membrane potential. And if we did, then we can initiate an action potential. By golly, folks, we hopefully will do it. Well, what do we need for that to happen? Okay, we need a minimum value. Okay, I won't say it's a very small value, right? But the minimum value, because that's obvious, because they call it minimum, right? We need to have this minimum voltage value to be reached. That is, all right, what our threshold value is. It's absolute. Okay? The threshold value is negative 55 millivolts. Our resting membrane potential is negative 70. So the difference between those two numbers is 15, but it's a positive 15 because we're moving in a positive direction. We are making, all right, our value more positive. Okay, negative 70, negative 55 threshold value. That's a difference of positive or plus 15 millivolts. That's the minimum voltage change that we need. So we have to generate enough EPSPs, because they're excitatory, to reach that value. Now, meanwhile, if we're just generating EPSPs, that's not too hard. But keep in mind, we might have some other presynaptic neurons that are generating IPSPs, which will then cancel out EPSPs. So we need more EPSPs ultimately than IPSPs, and they have to reach this plus 15 millivolt value. That's what we need to do. So if that occurs, and that happens in the initial segment, the axon hillock, then guess what? We are going to start the generation of an action potential, and we'll open up our voltage gated channels, because we don't have any voltage gated channels in our receptive segment. In our conductive segment and in our initial segment, we have voltage gated channels. So now we'll see the opening of these voltage gated channels, and now we will have done what we've been wanting to do, and that is to create an action potential. Our very own nerve signal. We've done it, everybody. We did it. So when we talk about summation, this occurs, I always love to say this, across time and space, or space and time. Basically, right, when we're adding up all of these IPSPs and these EPSPs, we're going to include all of those throughout the dendrites and the soma. And this occurs over a period of time. So those IPSPs, all right, it's a one-to-one negation factor, okay? One IPSP will cancel out one EPSP. So there's two ways in which we'll reach summation. There's spatial summation and temporal summation. That's easy. Spatial summation is space, okay? And temporal summation has to do with time. Okay, so what we'll see with spatial summation is that we're going to have, all right, neurotransmitter being released by all these presynaptic neurons, all right, all over the receptive segment of our postsynaptic neuron. And so this will happen, like I said, simultaneously. So some of those could be IPSPs, some of those could be EPSPs. It's going to look something like this. You saw this picture before. This is spatial summation. This is showing you that some of those presynaptic neurons are generating IPSPs and some are generating EPSPs. And so what's going to happen? We're going to hopefully add them all up at the initial segment, and we'll get that minimum plus 15 millivolt value, and we'll reach threshold. Now, temporal summation, different story. That's over time. And that's when we're dealing with one single presynaptic neuron, but it just keeps like rapid fire, right? Just repeatedly releases its neurotransmitter, and it will produce several EPSPs for a quick blast of time. So here's the spatial summation. Okay, here it is again. And so you can see all these different neurons here generating their miniature graded potentials there. And they just add up, one on top of the next, on top of the next, on top of the next. And so it gets us ever so closer to the threshold value until We get that threshold value in the axon hillock, and bam, we have an action potential. Here's the temporal summation. Okay, so the temporal summation we're going to see here. One single, all right, presynaptic neuron is repeatedly, like a Morse code person tapping on one of those telegraphs, right? It's just repeatedly releasing neurotransmitter, all right, onto the receptive segment. And each time it releases the neurotransmitter, it gets closer to the threshold value. And it does it just so close to one another. And that's what we see there. And that's our temporal summation. So this brings us now to the third segment of our neuron. Oh, no, wait, I'm sorry, not the third segment. All right. Still in the second segment. I'm getting ahead of myself. That's the conductive segment. But this brings us to the all or none law. I love this law. It's very simple. Okay. You either have it or you don't. That's basically what it's saying. So once we reach that threshold value, once we reach that negative 55 millivolts and our voltage gated channels open up and we start to generate that action potential. Here's the nice thing. Once that occurs and the action potential is begun, you can't stop it. Okay. It's going to travel from point A all the way down to the very end of the neuron to point B. And the beautiful thing about this is it does not lose any intensity. Now we saw that graded potentials did. They lost intensity. Okay, but with the action potential, all right, we don't have that. We don't see a loss in intensity. We'll talk about more of that in a moment here. All right, but if the threshold value is not reached, okay, our negative 55 is our threshold value. Say we get to negative 60, we're close. Negative 57, we're still close, but we're not at negative 55 or less. So we reach what's called sub-threshold value. The voltage gated channels do not open up, they stay closed. And if they're not opening, then guess what? No action potential. Not happening. Not happening. The all or none law also does state that because you do not lose any intensity, we will see the same intensity throughout the entirety of the axon. Okay, so it doesn't matter, all right, if you get to just barely negative 55 or if you blow past negative 55 and get to like negative 40 or negative 30 or negative 20. Okay, it doesn't matter. It's like pulling the trigger on a gun. You can pull it hard, it's still going to fire the bullet at the same speed. You can pull it softly and slow, right? It's still going to fire that bullet at the same speed. It does not matter. Okay, so it doesn't matter how you get there. Okay, it matters that you just get there, all or none. What is the significance of the threshold membrane potential in the initial segment of the neuron? All right, this just talks about what I just mentioned here. Okay, threshold value is when we initiate the action potential. All right, sub-threshold value is, hey, good try, you got close. No participation trophy or any type of trophy for you because you didn't generate the action potential. All right, and so you get nothing. It's all or nothing. Okay, so if you got threshold value, better luck next time. All right, so the conductive segment. The conductive segment is the axon. And specifically, when we're going to be talking about these action potentials and what happens, we're going to be talking about the plasma membrane of the axon, and that's called the axolemma. All right, so what is an action potential? Yes, it's a nerve signal, but what does it really incorporate? It incorporates the depolarization of the plasma membrane of the axolemma and the repolarization of the axolemma. So we're going to see how for a brief period of time, okay, a resting membrane, which is normally positive outside and negative inside, we're going to see how we flip it. We're going to, during this action potential, we're going to make the outside negative and the inside positive for a brief moment. Okay, so that's what we're talking about, the temporary reversal of the polarity across the PM, which is the plasma membrane. All right, so we know what depolarization is and repolarization, right? We make the cell, all right, more positive with depolarization. And with repolarization, we return the resting membrane back to that -70 value. So what actually happens during depolarization in the axon? Well, right, the voltage-gated sodium channels open up and sodium enters into the cell, bringing its positive charge with it. So it makes the inside of the cell more positive. Repolarization, okay? What happens then? During repolarization, again, we see the opening of voltage-gated potassium channels. And so when they open up, potassium leaves the inside of the cell, taking its positive charge with it. And it makes the inside of the cell more negative as we get return back to our resting membrane potential. And so this occurs all the way down the axon to the synaptic knob. So you'll have all these channels, these voltage-gated channels next to each other. And so one will open up, and then that will trigger the next one to open up, and that will trigger the next one to the second one to open up. It's like dominoes. When you stack them up and then you push them down, that's how this works. You get the sequential opening of these voltage-gated channels all the way down the entirety of the axonal lemma there. And so that will generate that action potential, which we call a nerve impulse or nerve signal. Okay, that's what we're gonna see. So now I'm really kind of excited because these are new slides for me. So I've never taught with these before. All right, so I'm gonna walk you through. Probably won't finish all of this, okay, but I'm gonna walk you through, right, how we are going to generate an action potential. So it's always starting off with a resting membrane potential, meaning our resting membrane potential is that -70 here, okay? So now we're going to talk about how we first start off. Remember what I said, an action potential is depolarization followed by repolarization. So we have to depolarize this cell, right? So how does this happen? Okay, well, we're in the initial segment. Okay, we're waiting for sodium ions from the cell body, right, to change the membrane potential near us. Okay, so what happens is, right, these EPSPs are coming along and they're bringing sodium ions with them, and they're causing those sodium ions to diffuse into the cell, and they travel, right, parallel to the plasma membrane. And so they get close, all right, to where we're at, because we're sitting right there on our first voltage-gated sodium channel. Right? And so that sodium enters in, and it changes that value now of the membrane. All right, it goes from -70 to -55. And that was our stimulus. That -55 got us to that threshold value. And as soon as that happens, as soon as that happens, right, the activation gate on our voltage-gated sodium channel swings open. Well, you guys know the rest of the story, okay? As that gate opens up, right, sodium is now going to flow through that open channel, going down its concentration gradient. Also getting pulled in to the inside of the cell because it's negative inside there. Well, only for a brief moment, because so much sodium is rushed into the cell, right, and it's brought its positive charge with it, it has changed now the membrane potential to +30. Look, you can see here, it's positive outside, negative inside, before any sodium moved. Now, with all that sodium moving to the inside, it's made the inside of the cell positive and the outside negative. So we have now reached this +30 millivolt value here. Okay, so that's what happens on our first two steps. Now, right, our voltage-gated sodium channel is going to enter into the inactivation state. Remember, there's 3 states to the voltage-gated sodium channel. So what we'll see is, all right, here Once we get to that positive 30 value, the inactivation gates slam close. Because remember, voltage gated sodium channels have two gates. The activation gate, which is that little swing arm, and then these swellings here, that's the inactivation gate. So the inactivation gate momentarily just slams shut, preventing any more movement of ions. Okay, brief moment here. All right, and it's good that this occurs, and I'll explain to you later, all right, why that's relevant when we talk about the refractory period. All right, so while this is going on, right, we're going to see in an adjacent region to us all of this stuff happening. Okay, remember, I told you this is a sequential series of events, right? So it happens like dominoes falling. So one gate will open, and then the next gate will open, and the next gate. And so that's what we're going to see here. Okay, so as this gate closes, all right, we're going to see this next gate here, all right, start to undergo the same thing that this gate underwent on the previous slide. All right, so we're going to stay here with this guy. So we're at that positive 30 value. Okay, so now our voltage gated sodium channel is closed. And so what we see now is we see the opening of our voltage gated potassium channels. Once we get to that positive 30 value, that triggers the opening of these voltage gated potassium channels. And so potassium then starts to diffuse out of the cell, going down its concentration gradient. And so it brings its positive charge with it, and it makes the outside of the cell positive, and the inside of the cell returns back to its negative value. You can see as it starts to fall down. And so we're trying to get back to this negative 70 resting membrane potential. Okay, but unfortunately, the voltage gated potassium channel was left open a little bit longer than we intended it to. Or maybe a chloride channel was open and chloride ions had moved across the plasma membrane. Okay, but what we see is it stays open a little bit longer for this fella, and a little bit extra sodium leaked out or got out of our cell, and we overshot our value of negative 70, and we jumped to negative 80. But this only happens briefly. Okay, we hit that negative 80 mark. Right, all of the voltage gated potassium channels will be closed at that point. And so no more potassium will leave. Okay, our voltage gated sodium channels are still closed. So what happens? Okay, what we'll see is our voltage, not our voltage, excuse me, our sodium potassium ion pumps will still be working, and they'll bring us back to that negative 70 value there. And so this will repeat. All of this is going to repeat, all right, as we move down the axial lemma. All right, this is a perfect place to stop. I don't want to, folks. Trust me, I really don't. But it's a perfect place to stop because